Good evening and greetings to one and all gathered here. It gives me an immense pleasure and warmth to welcome you all to this Biotalk series version 2.0. Last series, last week we had series version 2.1 on the topic Impact of Herbal Medicine in COVID Management. Today, in our midst, we have an eminent speaker, Dr. Sirujunisa Abdul Razak, who is going to give a speech on the topic nanotechnology in food preservation. Now I call upon Ms. Kalavadi to give welcome address.
Thank you, Alfred, for your invite. Good evening, everyone. On behalf of the Department of Biotechnology, St. Joseph's College of Engineering, I take immense pleasure in welcoming you all to the Biotalk series version 2.2 on the nanotechnology in food preservation. It is indeed a great pleasure for me to welcome the speaker of today's session, Dr. Sirajanusha A., a postdoctoral fellow from the Department of Food and Biological Engineering from Jiangsu University, China. I also extend my warm welcome to the head of the department, the faculties of the department, and my students. I once again welcome the speaker. You're welcome, ma'am. And now I request our head of the department, Dr. G. Srikumar sir, to present the inaugural address. Thank you all.
Thank you, Kalavati, for a good invite speech. Good evening to my dear students and staff members. Hope everything is going fine and safe. I'm happy to see you all again with this virtual BioTalk lecture series version 2.2. As a part of inauguration of this session, I wish to highlight some of the credits of our college and the department. St. Joseph College of Engineering, one of the top colleges in India, with the 160th position as per the current NIRF ranking. It has got 27 years of high reputation in educational society with the NBA and the NAAC accreditations. It is having 10 UG programs and 7 PG programs with an affiliation from Anna University, Chennai. Recently, we have received autonomous status also from UGC, which will be implemented from the forthcoming academic year. Our Department of Biotechnology is started in the year 2002 and having BTech and MTech programs. Since inception, we have bagged 139 university ranks with 10 golds and 7 silver medals and consistently maintained 100% pass percentage every year. The department has got sophisticated laboratory facilities and fetched a research center recognition from Anna University to do PhD program. We have more than 1.2 crores of government funding for conducting research in our laboratory. Current COVID outbreak and the subsequent lockdown makes us inert by both physically and mentally. This problem is more realized especially by the students and academicians because all the time we were mentally more active when compared to others. In my view, high priority should be given on making our biotech community to be abreast with the current ongoing research trends and not allow them to lose their interest. Our department has the practice of conducting BioTalk lectures series, and this comes under that version 2.2. And by bringing eminent personalities through webinar and sharing their research views to the students, teachers, and research personnel. Today's lecture topic is going to be nanotechnology in food preservation, which is a highly application-oriented field. United Nations report says that yearly 931 million tons of food being wasted globally, and the food waste in India is around 68.7 million tons per year. One of the main reasons for this wastage is inadequate preservation techniques. If we develop suitable food preservation method, we can decrease the production cost and increase the efficiency of food usage, improve food security, nutrition, and contribute towards environmental sustainability. Nanotechnology is the recent technology that use matter at an atomic, molecular, and supramolecular scale for catering the industrial needs. In order to give more authentic research-based explanation about the use of nanotechnology in food preservation, we have Dr. Sirajunisha A, who is a postdoctoral fellow in Department of Food and Biological Engineering, Jiangsu University, China. On behalf of the patrons, Dr. Babu Manoharan, Chairman, Madam Jessie Priya, Managing Director, and Mr. Sasi Shekhar, Director, Dr. V.S. Rao, Principal, and Madam Parvatavardhini, Dean. I welcome the speaker, Dr. Sirajunisha, and all the participants to this BioTalk lecture series. I also convey my warm felicitation to Ms. Yuvarani and Ms. Kalavati, who is the assistant professors of our department, for organizing this webinar. All the best to you all. Now I ask the student coordinator, Mr. Alfred Francis, to give a brief introduction about the invited speaker. Over to Alfred.
Thank you, sir. Dr. Sirajunisha has completed a Bachelor's of Science in Microbiology and Master's of Science in Biotechnology from Bharatiyar University. And then she completed a Master of Technology in Biotechnology from Satyabama University. Later on, she went on to pursue a PhD in Chemical Engineering from Annamalai University, funded by Non-ASAP Meritorious Fellowship, UGC. She has worked as a lecturer in Andar Aragar College, Chennai from 2009 to 2010, after which she pursued two postdoctoral fellowships, one from Anna University and another from Thailand. Currently, she is a postdoctoral fellow in Gyeongsu University, China. She has published around 22 national and international papers. She has also published 3 book chapters and currently 6 articles are under review. She is a recipient of many awards from various institutions and research centers. Her main fields of research are exploring the diversified capabilities of nanoparticles from natural resources in environmental and health, and the use of microalgae in producing renewable energy and food preservation, and also fabrication of natural polymer to degradable films and for biological applications. Ma'am, I kindly request you to take over the session. Thank you.
So good evening, everyone. I guess I'm audible to everyone.
Yes, ma'am.
Yes, ma'am.
Yes, ma'am. Yes, ma'am.
Thank you. So I'm overwhelmed by the welcome given by Dr. Shri Kumar, the professor and head of the Department of Biotechnology, and thank you for inviting me for this lecture and being good organizers. And this is to Ms. Kalavati and Ms. Yuvrani, the assistant professors, and to student friends. This is a little talk on nanotechnology in general. What do we do in food preservation? And this is basically for you to understand the basics on food preservation and the role of biotechnology in preserving foods, and the way the pathways, the openings where you can work in for commercialization or to undergo or pursue research. Okay, this is for you only, students. So this is basics, and just you can go through whenever you want. So let us start with the
Yes, ma'am. Your screen is visible. Yes, ma'am.
Okay. So we'll talk about nanotechnology and food preservation. So the slide itself, just for your understanding, better understanding, the slide itself shows a polymer film with various nanoparticles impregnated in it, and one of the most important nanoparticles that we use in food preservation is nanoliposome, which is a bilayer of lipid layers, and with studs of proteins or any molecule that you want to incorporate into food, into your human body or into the environment. So this is the overall picture of what we are going to study. Okay, so let us give a short introduction of the slide of the show today. So due to modernization and due to heavy work, the mechanization of the globe, we are into a faster pace of working. So we go in for packaged food more than the normal food that we eat with all peas at home. And due to that, there is increase by 60%. That there will be an increase by 60% by 2050, so that you get more food in a rapid manner. Okay, so due to that increase, food researchers and other biotechnology people and technology people who work with nanosciences, they keep forcing themselves to identify alternates and identify different substitutes with nanoparticles to ensure proper food production and to meet the demands of consumers. So as our professor just now said, there are millions and billions of tons being wasted every year without being consumed, or because of the failure of storage, because of the failure in transportation facilities, etc. So in order to maintain that, we need to find an alternative or a replacement to sustain the shelf life of the product, and how not to waste food by via consumption. Okay, that is one objective of food technology. Next, another objective of food technology is how can you protect the environment. That is, after you consume the food, you keep throwing the papers, you keep throwing the covers, yeah, on the non-degradable plastics. That is mainly from polyethylenes, like the polyethylenes, polyesters, polyterphthalates, etc. So those synthetic plastics cannot be degraded by the soil microorganism or the soil creatures. So that has to be avoided. And what WHO says, that is World Health Organization says, is more than 20 to 200 illnesses are caused by only the petroleum based plastics. So we have to stop that also, and that has to be replaced by natural sources, and natural sources are plants, animals, and microbial sources. So these also produce polysaccharides in the form of cellulose or starch or animal protein, animal starch like chitosan. Microbes produce as capsular or slimy polysaccharides, as extracellular polysaccharides. So these can be made use of for producing pure bioplastics or blended plastics. Okay, and where nanotechnology comes into play is in restoring the polymer, the mechanical strength and the physical strength of the polymers. So when we incorporate nanoparticles, they get stronger. There's no movement of polymeric strands, and that gets mechanically strong. So these nanoparticles also play a role in protecting from microbial contamination. They protect from moisture. They protect from different gases being released from the food or entering the food or entering the packages. So these are all the objectives that we are going to clarify within this talk. Okay, now we'll start with what nanoparticles are, what nanomaterials are. So nano, it is 10 power minus 9 meters. So those particles which only range between 1 to 100 nanometers are suitable in food preservation. That is, those are exactly the nanoparticles, and those particles which lie within are suitable for food preservation, and that can be incorporated inside the polymers. And one of the most important nanoparticle which is currently in trend are quantum dots. Okay, you should have heard about quantum dots. So that has very minute nanometric size, that is from 2 nanometers to 10 nanometers mostly, and they have a very distinctive feature of exhibiting fluorescence. They transport electrons, they excite electrons, and they transmit fluorescence when there is passage of UV radiations. Okay, and that nanodots have a tiny core, which is the main substance of fluorescence. Then that is covered by a protective layer called shell, and when you use it as a nano carrier, or when you use it in the food packaging system, that will be that will have extra secondary molecules like proteins or lipids, whichever that should be imparted into the human system as food or protecting drugs. Okay, and so these nanodots are now in trend. This you can, for the students I'm saying, this you can take it up for research. Okay, and this can exhibit all the colors in the EMR. So based on the size of the quantum dots, if it is 2 nanometers, then it reflects blue light. Then if it is above 5 nanometers or 7 nanometers, then it emits red light. So it is based on the size of the molecule, size of the quantum dots. They are the smallest materials used in detection of diseases or protection of foodstuff. Okay, and the other nanomaterials that we use are in different dimensions. It is 0 dimension, 1, 2, and 3. Okay, based on their dimensions, they are called as nanoparticles, quantum dots, clusters, and based on the shape, they are called as rods, dendrimers, capsules, whiskers, etc. And when they are clubbed together, when nanoparticles, when 0 dimensions or single dimensions are clubbed together, they get agglomerated as aggregates, and they form 3-dimensional particles. And in food research, we use nanocomposites, and they are polymers with nanomaterials. Okay, the polymeric strands impregnated or incorporating nanoparticles. So they form nanocomposites, and that can be further used as packaging films. So why do we have to exactly go for only nanometric materials? Because they they have a larger surface area. Why do they have a larger surface area? Because they have their exact surface atoms exposed. Okay, so the undesired moieties present on the surface of those nanomaterials, when they reduce in size, or through reduction process or oxidation process, or by reducing the size via mechanical shearing, you have the moieties getting neglected and more of the surface gets exposed. Okay, and those surfaces will be exposed for the other amino acids or any molecules of desire can be attached. Okay, so the specific larger surface area is one of the main properties for these nanomaterials being used in food research. And when mixed in polymers, they'll exhibit more strength. Like they can form bonds very easily, and the bonds are very stronger. That will be mostly electrostatic forces or any van der Waals interactions. Another advantage of using nanomaterials in polymeric films are they can induce crystal polymorphism. when there is when you impart nanomaterial to the polymeric surface, they tend to they tend to slightly change the polymeric structure of the polymeric structure, and that helps in enhancing the mechanical stability of the packaging film. Okay, and this also helps in increasing glass transition temperature. That is, it can change from the temperature that helps in changing the glassy state to rubbery state. That gets increased, so we can use nanomaterials for mechanical stability and physical durability also. Other improvements that that is given by nanomaterials to a packaging film, okay, are enhanced tensile strength. There will be reduced elongation at break. There will be higher thermal stability. There will be no the resistance against gases, that is entry and exit of gases, will be more. Then you can and other common advantages are can be recycled, they can be degraded, they can be renewed also. If you are using magnetic nanoparticle, you can recover it and you can repeatedly use it for the process of production of preparing packaging materials. So why I give stress on packaging material is that is the main source of entry of any contamination. So more research in food sector is going only on how to create a sustainable packaging material for increasing shelf life and much of durable material. Okay. So in daily life, we keep intaking natural nanomaterials. Like, you eat whipped cream, and that is a nanomaterial. You eat custard, that is a complex of nanomaterials. You eat ghee, you eat butter. Those are all emulsified products, and they are completely made of particles of nanometers. Okay, and certain milk proteins and casein, different starch materials, the shells of certain people who eat animal foods, they eat prawns with the outer covering, and that is that material also is considered as nanometric product. Okay, so in daily life, without knowing or knowingly, we take nanomaterials. So these nanomaterials, since they are completely sensitive to environment and that can be degraded very soon, we don't use these nanomaterials in packaging systems. We fabricate in laboratories, we do in vitro, and then we go in for using it as nanomaterials for packaging and processing. Okay, we get into the topic now. Why nanotechnology in food sciences? How nanotechnology play a role in food sciences? So from this slide, you can know that the nanomaterials role starts from agriculture, from land till it reaches humans. Okay, till the product reaches human. So in agriculture, you can create nanomaterial, you can create new pesticides with nanomaterials. By incorporating nanomaterials, you can target the contaminants using nanomaterial coated, using pesticide coated on nanomaterials. Okay, because nanomaterials have the role of controlled drug release and targeted drug delivery. Okay, only when there is any contaminants attacking the plant or attacking the soil, then this nanoparticle identifies the organism by electrostatic force of attraction. They go bind. If any receptors are perfect, then they start degrading the organism. Until then, it will not let the pesticide inside the plant or on the soil. So pesticides will be pesticides without any work. So random effect of pesticide on soil or plant will usually kill. usually have adverse effects. So if you coat it on nanomaterials and spray it on the soil or inject it into the plant, they stay until contaminants arise. Okay, and you can deliver agrochemicals also. When whatever desired to the soil can be delivered as what I said now. Then it can sense any, if there is any contaminants on the soil, it can also sense. So we can use nanomaterials as biosensors, and how biosensors work, I'll say it later on. So this is in agricultural sector of food science. Next, the food is taken for processing, okay, to be delivered to humans. And there during processing, you can use them as, you can use nanomaterials for, as usual, for targeted delivery of nutrients as nanomicelles. Then you can use it as viscosifying agents, emulsifying agents. You can use it against caking, okay, where nanoparticles help in preventing moisture and gases, okay. And this product, and after processing, you send it as products, and these products need protection. So you can use nanoparticles in packaging. You can prevent nanoparticles. You can prevent UV radiations using nanoparticles. You can prevent microbial contamination. So hygiene is more imparted by nanomaterials. Then these products are given to human beings as nutrition, okay. And these nutrition, in the nutrition sector, it can be used as fortifying agents, okay. So in biscuits, you can impart iron nanoparticles. You can impregnate iron nanoparticles and give it to anemia patients. And you can create more fortified foods for any patients who cannot directly take that particular mineral or vitamin, okay. And these, and in case of water purification, you can use nanomaterials for these nanomaterials, with the help of any light radiations, with any with normal white light or any UV light or any other radiations, it can treat the it can treat the water by breaking heavy metals or any pollutant via redox reactions. So this is the general outline of how it is being broadly used in food sector. And when you get into food industry, how does it work? How nanotechnology works? Okay. So first of all, it is used as materials. Just as nanomaterials, you can mix it up with food as nanomaterials or nanoemulsions or nanocomposites or any nanostructures. Just for it to be used as direct nutrition, for it to be used as nanocarriers, or it can be used as agents that prevent food contamination, and it can help in desiccation of, it can maintain the food in the desiccated state without any moisture for a longer shelf life. So these, after you add particles, then you process the food, okay. And during processing, we check the rate of reaction with the product, we check the mass transfer phenomena, we check the heat transfer phenomena, and that happens during the processing of food. Then you bring it out, bring the food as products, and that product. So when the food is as product, nanotechnology plays in the role of formulation, packaging, and delivery, where if any extra formulation, if any nanoparticle has to be added in food after you process, that is as desiccant. If you want it to be added as a desiccant, you make a sachet of nanoparticles and put it into the packet, okay, packet of processed food. Then you pack with a packaging film incorporating nanomaterials. Then you deliver. When you deliver, you add barcodes. Okay, and those barcodes will identify whether the product is safe or it is getting decayed. Then, yeah. So after you deliver, after the product is out of the out of the industry, it has to be delivered to the market. Till that time, the transportation time, you need the food safety and security to be eyed upon. So for that, we will be using nanosensors and nanotracers. So here we'll be using indicators, temperature indicators, pH indicators, etc. And that will be nanoparticles which is coated with any uh, what do you call, any pigments or any indicators that will change colors or that will change its own, that will indicate by light. Okay, so based on the color change or the light, the emission of light, you can sense whether the food is being decayed or it is being damaged. So different kinds of nanomaterials are involved in food science. Okay, so there are various types of nanomaterials other than these being displayed also. There are nanoparticles which are used, and that will be mostly metals or metalloids, and it will be used against microbial contamination. Then nanotubes, nanodendrimers, nanotubes, nanodendrimers, these are all used as materials that helps in retarding the diffusion of gases. Okay, and nanoclays also. Then nanomicelles, nanocapsules, nanospheres, nanoliposomes, all these help in encapsulating the nutrient that you want to deliver. The nanosieves help in preventing the entry of microbes because because the porosity will be in nanometers. So nanosieves help in filtering the pathogens from atmosphere. Nanocochleates are spiral cigar-shaped nanomaterials where the drugs are incorporated within the spirals of the nanocochleates, and that is also used in drug delivery and nutrient delivery. So nanolaminates, nano composites, they help in they are used as packaging films, and quantum dots, they are free features of nanotechnology. Okay. Yeah. So this table I have made is made for you to know about what type is specifically used in which sector of food science. Okay, and nanoliposome is generally used in encapsulating vitamin A and iron. Vitamin A is generally vitamins are very sensitive to environment. So when they are randomly sent in, it can be damaged due to different pH or different temperatures within the human system or in the environment. So that has to be encapsulated for better delivery. So we'll encapsulate vitamin A and iron in nanoliposomes, and nanomicelles and nanocapsules for oils. Okay, oils generally for cholesterol patients or any cardiac patients, we cannot directly they cannot ingest oil directly, so we'll be encapsulating within nanocapsules and nanomicelles. Nanoemulsions, emulsify, they are given as so we keep whipping it up to produce a colloid of emulsion, and that will be toffees. That will be mainly used in toffees, margarines, cheese, and ice cream industries. Okay, so nanoparticles and nanodots, they are used in toxicological studies where you check whether the product is releasing reactive oxygen species or whether it is releasing any gases because of decay, or whether it is exposed to any microbial organisms which cause pathogenic results. Nanoclays are generally used in as I said before, it is used used as a barrier of gas diffusion. Nanosensors, it is used in food safety and security, where it can detect even it can detect earlier, before even it gets decayed. It can detect the decay of food stuff, and it has also the ability to detect whether the food is wild type or genetically manipulated food. Then nanofibers, nanolaminates, nanocomposites are used for packaging, and they are used in nanofibers are used as biosensors also. And nanocochleates and nanodroplets, they are used as aerosols, mainly as vitamin sprays, where one is allergic to any nutrient, or when one is not able to ingest a nutrient, we can give it as aerosols or sprays. Okay. So different particles play a role in preservation as in different ways. Okay. Silver nanoparticles, it just gets into a bacterium. It can, silver nanoparticles, they are always advertised as materials that stop pathogenic contamination. It's because they're able to, because of their nature, because of these ions nature, that is positively charged ions, they get bound to the negatively charged cell wall of the organisms, and they just straight away damage the plasma membrane. They they destroy the channels of the cell wall, and they get into the pathogen, into the bacterium, and they break open the nucleoid, that is the nuclear material of the bacterium, and thus destroy the organism. So everywhere silver is being used, silver nanoparticles are used against pathogen contamination. And clay nanoparticles, mostly clay is used for filtration. In any industrial sectors, in industrial production, clay nanoparticles or clay as such is used as filtration materials. And here as nanoparticles, clay as nanoparticles is used in plastic bottles, where they stop diffusion of gases, so that carbon dioxide, if there is any permeability of carbon dioxide or oxygen, say in case of beer bottles, we need carbon dioxide more. That carbon dioxide has to be inside, and that should not be diffused. So we put in clay nanoparticles along with the plastic polymers, and that will retard the diffusion of carbon dioxide, and it will not allow oxygen also. So it has dual purpose, and hence this clay nanoparticles will be added to beer bottles. And clay in the form of kaolin, or I guess it is it is mostly kaolin, I guess. Yeah, that is used as nanomaterial. Then regarding nanomaterial intake directly as nanomicelles. These nanomicelles will have the will have the nutrient inside the single layer lipids, and that when you intake, based on the pH that remains on the tongue and the stomach, it gets discharged. That is, if the nanoparticles Yeah, if the nanomicelles are larger, then it straight away disintegrates, disintegrates in saliva. If the micelles are very smaller, then it can be ingested, and that will break open in the stomach based on this is mainly based on the pH. Okay, so the current trend in food technology is use of nanocomposites in packaging films. Okay, so many researchers are only concentrating on packaging films. So in order to cumulatively protect the foodstuff. So packaging film is composed of hydrocolloids, plasticizers, fillers, functional additives, and cross-linkers. It is mainly the polymer and the nanoparticles. Plasticizers, fillers, additives, they are all, the secondary molecules that are needed for preparing packaging material. So the main product is hydrocolloids, that is the polysaccharides in water or in any organic acids. Okay, and that you can obtain, this is to eliminate the use of synthetic plastics. So natural polymers are available in markets as carboxymethyl cellulose, cassava starch, gelatin, pectin from fruits, alginate carrageenan are from algae. You can use algal polymers also, which is more appropriate. Chitosan from prawn shells, crab shells, etc. Then soy protein can also be, in rare cases, used as hydrocolloids. Okay, and in certain cases, when you use pectin as the colloid, as the natural polymer for production, it also needs binding agents. So these binding agents can be any of these polymers. Then plasticizers. What are plasticizers? What do they do is, when there are strands of polymers, okay, it needs integrity. It needs stability. For that, into the free volume, these molecules, glycerol molecules or sorbitol or invert sugar or corn syrup, they'll go bind within the strands, in between the strands, and they maintain the stability. And these plasticizers help in maintaining the flexibility also. They do not, you can theoretically say it increases the grip of the polymer strands, but it is not so. It gives a smoothness to the packaging film, and it increases the flexibility and stretchability of the polymeric film. And above this, you will be adding nanocomposites also. It acts as plasticizers, as binding agents, as cross-linkers also. It helps in, maintaining the integrity, and helps in, increasing the mechanical strength and thermal properties. Also, it protects the particles from food from pathogenic contamination. Okay, so as we discussed earlier, nanocomposites are made up of multi-constituents of various dimensional nanostructures, like nanoparticles, nanocrystals, nanoclays, etc. Okay, and you can also add functional additives. So functional additives are nothing but phytoconstituents, like secondary metabolites you would have studied in plant biology. So secondary metabolites like alkaloids, terpenoids, flavonoids, etc., phenolic acids, tannins, etc., which gives which gives extra protection to the food stuff that is being packed. Okay, and this nanocomposite, when it acts only as nanocarriers, additives can be nanofillers only will act as protecting agents. So there is no need of adding additives. Only mostly when you add metals or metalloids, which by itself do not much impart protection to the food stuff, you can use phytoconstituents. Then cross-linkers. Cross-linkers are not much needed when you add nanocomposites, but in case in exceptional cases you need cross-linkers, and that will be mostly mineral salts, enzymes, amino acids, and organic acids. So when you're packing a food, you need to take into consideration the mass transfer phenomena. That is nothing but when a layer of packaging film is being put upon a foodstuff, there has to be headspace for it to breathe in and breathe out. That is, for it to release gases, okay, and for it to maintain a proper environment. So, and it has, and this nanomaterial, this packaging material should not let any environmental gas, any gases from the environment, and it has to have the ability to protect the food from moisture and pathogen. Yeah. So you need to learn the rate of permeation, how the volatile gases are being are entering and exiting, how whether the packaging film has the ability to prevent absorption from the foodstuff. That is, whether it can prevent the absorption of fatty acids or any volatile matter from the food, or any pigments or vitamins from the foodstuff. You can see, very not packaging materials of non-standards. They on due course of time they start acquiring the pigments from the food, and these materials will have the pigments. So that is not a good standard of packaging material. So that has to be studied always. And migration of volatile compounds is also to be noted. So that could be learned from the diffusion rate. So permeation, absorption, and diffusion are certain phenomena which has to be studied and taken into consideration, and that has to be kept under control. Otherwise, the food might lose its flavor, aroma, or any desired products, and it can be prone to infections. Okay, this is for the research purpose in the laboratory. So just for the students, how do you prepare a nanomaterial? How do you prepare a packaging material? Okay, so you have to prepare a mixture of colloids. If you need, if only you need plasticizers and cross linkers, you can add, or else just go for the colloids for the polysaccharides in water or organic acids. Chitosan will be dissolved only in acetic acid. Other polysaccharides can be dissolved in water. So you dissolve it, you homogenize it under a magnetic stirrer. If you want any phytoconstituents directly, you can also add that, or else don't add. You can just cast it. Casting method are of two types: bench casting and continuous casting. Bench casting is just pouring it on a on a tray of known dimensions. Just pour, let it dry. After it dries up, you can detach it. You can take it out as a polythene sheet, and that is how we prepared in the lab. And continuous method of casting is, it is in industrial scale. That is, they keep pouring it on a conveyor belt, and it keeps rolling. It gets into a heating machine, and on the other side you get the sheet. Okay, so that is happening in industrial scale. So this is a normal way of producing biopackaging film, and if you want to mix it with the nanoparticles, first you have to prepare nanoparticle in the laboratory, and that is a whole set of study. So just I'll mention how you prepare nanoparticles in the lab. So you take precursor molecules, precursors like if you want to prepare zinc oxide nanoparticles, say for example zinc oxide nanoparticles, then you'll be taking precursors like zinc acetate or zinc sulfate or zinc nitrates, and then you'll be adding a reducing agent to it, say boron anhydride, sorry, borohydrate, no? Yeah, sodium borohydrate and or sodium hydroxide or potassium hydroxide, which has hydroxyl molecules. for it to act as reducing agents, and that reducing agent will act upon the precursor molecules, and upon redox reactions you get nanoparticles. So here I have shown my iron nanoparticles, so that we can mix it with hydrocolloids and you homogenize it. So you go with the normal process, casting, drying and detaching, and you get the nanoparticle impregnated with packaging material impregnated with nanomaterials. So the difference will be bioactive film, the normal bioactive film, the common bioactive film will be a little thinner and it will be a little brittle, but nanocomposite bioactive film will be a little sturdier. So these are the nanomaterials we prepared in our laboratory. So normal bio packaging film via casting method from only algal polymers, that is carrageenan, sodium alginate. The next picture shows a polymeric film with nanomaterial coated. Here we have coated, I guess, zinc oxide. Next is just a common bioactive film only from pectin of fruit peels. No, the agri waste we collect from fruit juice shops. We extracted pectin from it, the crude pectin, and along with that we added algal polymers as binding agents, and then mixed it up to form a biopackaging material. Yeah, so this is how the nanopackaging material works. So once you cover the fruit, here I have shown fruit as a food. So when you cover food with the bioactive material, this bioactive material via electrostatic force of attraction, the microbes will bind to the polysaccharide, the polymer, and once the organism is attached, this nanomaterial, this nanoparticle starts releasing the phytoconstituent. Okay, so this phytoconstituent then will attack the microbial organisms. There are two ways of eradicating microorganisms. It just bioactive packaging material just repels the microbes or it kills the microbes. Okay, so based on the force of attraction, the bioactive packaging works. And you can either use a polymeric film as packaging material, or you can also directly coat the fruit or the meat with the hydrocolloids. Okay, so the picture shows like immersion. You have to immerse the fruit inside the hydrocolloid with nanomaterials, hydrocolloid containing the nanomaterials. You have to dip it in, take it out, dry, and then it forms a coat. So when you eat, you can just directly eat, no problem. You need not detach, you need not remove the coat. Yeah, how the nanoparticle in a biofilm works as an antimicrobial agent or an antioxidant agent. So first is food. Food encounters a microbial attack. Now food is being packed with packaging film containing nanomaterial. So based on the electrostatic force of attraction, the the organism binds to the nanomaterial. At that time, when the nanomaterial encounters the microbe, the metal ions from the polymer gets released. These metal ions then damages the cell wall. So it damages the cell wall, and all this happens under light. Okay, So and the cell wall gets lysed, and then it enters the cell. Okay, so first thing, destruction of plasma membrane. Then it enters into the cell. The metal ions then encounters all the cellular cytoplasmic inclusions like ribosomes, the other inclusions, etc. So these metal ions go bind to the ribosomal structures, and their dysfunction starts. And these nanoparticles also get into mitochondria because of their size. All these microbial agents, all the cytoplasmic inclusions, when they are when the porosity is micrometers, these nanoparticles, which are nanometers, they have easy penetration. Okay, so these metal ions, they penetrate into mitochondria, and they stop their functions. So there's no power, there's no ATP release. So there is, loss of energy, and that is how microorganisms die in one case. So when it is, when in the case of ribosomes, when they bind to the ribosomes, there's no production of RNA. If there is no production of RNA, there's no expression of proteins. So if there is no proteins, the cell is going to die. And then it goes bind to the nucleoid material. Nucleoid is, the nuclear material. Then it binds to the DNA, it intercalates with the DNA, and there'll be no replication, there'll be damage of DNA. So this is how it damages each and every part of the cell, internally and externally. And also all these causes cellular damage. Another thing is, it produces, in a chemical way, it starts producing what is that, reactive oxygen species like superoxide radicals, your hydroxyl radicals, etc. And that causes stress, and that induces apoptosis of cells. Okay, and this apoptosis is cellular, finally leads to cellular death. Okay, and exactly the mechanism of photocatalysis. What happens when light is, light plays a role in breaking of microbial organisms. So within nanoparticles, any atom contains valence band and conduction band. So from the valence band, electrons, once the radiations pass in, the valence band gets, there's excitation of electrons to the conduction band, and in the conduction band there is the conductivity happening. So these electrons, the accumulation of electrons, will bind to the atmospheric oxygen that is present on the surface of the nanoparticle. Now this oxygen molecules with the electron undergoes oxidation to form superoxides. Okay, and these superoxides cause apoptosis. Meanwhile, in valence band, after the negatively charged, these electrons go to the conduction band, here positively charged holes form. Now these holes will bind to, convert water to hydroxyls, and these hydroxyls also, being one of the reactive oxygen species, increases the oxidation potential, and that leads to apoptosis. So this is the way microbes are killed by nanoparticles in a packaging film. And once it is killed, the food is being protected. So this is one of the bioactive packaging materials where it is able to kill the organism only. So before even the food can decay, you can sense the you can sense the damage of the food. and that is by intelligent packaging systems. So intelligent packaging system is where you have materials, you have nano carriers loaded with any pigments or any materials that can change color or emit light. Okay, and here I have shown an example of anthocyanin. Okay, so now we'll take a nanomaterial, we'll take a nanoparticle, or we'll take nanorods, nanomaterial of your choice. We'll coat that with a phytochemical. Okay, and mostly pigments are indicators. They have the ability to change colors. So we'll be coating pigments. Say a natural, well-known pigment, anthocyanin. This anthocyanin has a cation named flavinium. That is that has an acidic pH. So when acidic, it is in red color. So when so you can see the product when you can see the packaging film in red color. As the days pass on, these fish meat or any meat, any beef or mutton or pork or fish, any meat tend to release gases. Okay, so release of gas is one prior information of the food getting decayed. So this gas will be detected by the anthocyanin on the nanocarrier. This colored pigment, red colored pigment, then gradually turns its color to yellow. That means there is a lot of gas being released, and one of such gases will be ammonia. So ammonia, when where it releases much, and it turns it into alkaline. And when it is alkaline, the color changes to yellow. And this yellow is because of a derivative called chalcone. So flavinium will be changed to chalcone. So acidic pH will be changed to alkaline pH because of the release of ammonia. So this is how intelligent packaging system works. So when it is fresh, it will be in one color. When it is, when it is getting decayed, the color gradually changes. So nanomaterials plays a role in smart foods. It is called smart food technique. Okay, so we have seen all the advantages of nanomaterials in food industry, but we have the character of just letting it in the environment. After you ingest the food stuff, what happens to the nanoparticles? What happens to the nanoparticles as the nanocarrier molecules that we have taken in? Or what happens after you eat, you throw away the material, you throw away the bio-nanopackaging material? What happens to the nanoparticles in that? Okay, though it is able to be degraded, certain nanomaterials cannot be broken apart. So what happens is there exists the phenomenon of nanotoxicology. So there is a permissible limit for each and every nanomaterial that can be ingested by the body. Otherwise, it leads to production of reactive oxygen species, causing oxidation within the human system and leading to apoptosis, and the human passes away. So in order to prevent oxidative stress, okay, the product has to be ingested it has to be taken in within the permissible limit mentioned by WHO. Otherwise, the risks will be heavier. So here I have shown a picture depicting the toxicity of nanomaterials in each and every internal organ. So mainly in brain, since the particle size is very small, it can penetrate the blood-brain barrier, and it can cause the damage to neuronal cells, causing neurological disorders. It can internalize, it can stay within the cells Okay, within the mitochondrion, within the cytoplasm, within the Golgi apparatus, within the ribosomes, and they tend to produce due to stress, due to the stress within the cells, they start producing ROS, and this ROS causes cellular death. Then, if nanoparticles go clog your alveoli, go clog your lungs in a broader spectrum, they tend to cause asthma, bronchitis, and lung cancer also. When it is into the you keep ingesting and it gets into the stomach lining or into the stomach system, into the gastrointestinal system, it can increase the acidity, it can increase pH, or it can decrease pH, or it can just penetrate into your microvilli and it can destroy the whole lining of the gastro system, and it can cause, it can start from inflammation, that is Crohn's disease, inflammation of bowel system. It can cause irritable bowel syndrome, and then it can also lead to colon cancer. So likewise, it can destroy heart, it can destroy the whole circulatory system, the nervous system, the lymphatic system, the skin, especially skin where it causes dermatitis, and it can also cause autoimmune diseases in dermal layers. So these are all the negative effects of nanoparticles if not taken in the controlled limits. So why do they cause, how do they cause toxicity to humans or to the environment? It is based on solubility. If the solubility, if the nanoparticles have very poor solubility, then it cannot be ingested, it cannot be broken down, it cannot be eliminated. The surface area. So if the surface area is larger, though it is a boon, it can also be a bane, surface area. So to that, when there is addition of any other molecules which are needed for the body, it will be eliminated along with the nanoparticles, and that destroys the human system. And depending upon the nature of the nanoparticles also, there is there is harmfulness to the body. And likewise, the chemical composition, the size, shape, the surface charges, all these play a role in causing toxicity to humans. So not only humans, here. So this is I've just mentioned certain reports that show the cytotoxicity, hepatotoxicity in human system. The main areas where you can find nanomaterials being settling are lung, liver, kidney. Okay, so intake of nanomaterial has to be properly noted. So in lungs, mainly in lungs, which is the easiest route of distribution, if the particle size is above 2.5 nanometers, they tend to stay in the nose, in the nasal tract. And if it is lesser than 2.5 micrometers, then they can enter the upper respiratory tract and cause bronchitis also. Okay, and if it stays, if it gets into and distributes within the blood system and finally enters the liver, it can cause hepatotoxicity. So increased use of nanomaterials cause damage to cells, human cells, leading to the final stages like final chronic illnesses like cancer and autoimmune diseases. So not only to humans, to environment also, nanoparticles are toxic. So how it causes toxicity is the same way. When it is exposed to light or in any illumination or any radiation, they produce reactive oxygen species by reacting with atmospheric oxygen and water molecules in the soil or in aquatic ecosystem. So these ROS causes cellular damages to soil creatures and microorganisms that are useful to the environment. So that has to be stopped. Then in the aquatic system, when fishes take in nanoparticles, these nanoparticles are generally not, ingested. They are not being broken down within the system of fishes. So they stay within their body, and it can lead to developmental changes if it is in their tadpole stage or in gestation stages, and it can then it can cause hereditary changes also. If the fishling produces, if the fishling is being produced by a mother fish, which has taken and transformed nanomaterials, these fishlings will also have the toxic effect of nanomaterials. So elimination or eradication or disposal of nanomaterials is in humans' hands after consumption. So to plants, the toxicity will be like it disrupts the carbon nitrogen cycle. So there's no photosynthesis occurring. I mean, there'll be no photosynthesis occurring, and hence the changes in plant ecosystem, terrestrial ecosystem also will be seen. Yeah, so these are all the toxic effects in the environment. So nanoparticles in food sector and to environmental and to humans, they are boon and bane. So when you use nanoparticle, when you do research in nanomaterials, please ensure that you learn the permissible limit of the nanomaterials that can be released into the environment, or that can be taken by the human beings from the World Health Organization guidelines. So out of this talk, we can conclude like food should not be wasted. Even if you waste in a proper way, that it is not being, it doesn't damage the food cycle or any human system or any ecosystem. So you have to use natural plastics. You better do research on natural polymers, prepare polymers which are needed for the environment with the nanomaterials that has no harm, that causes no harm to any of the creations in the ecosystem. So proper elimination protocol has to be designed, and you need to create, and a further step that is to be kept forward is creating a database management system. So that will have the note on different foods produced in the world, then the microbiota which are present in the world, and how the trade systems are happening within each country, so that the food sector will be, the foods can be transferred properly, and that can be consumed properly in a better way, in a harmless way. And the disposal, the final way of protecting the environment is the disposal of plastics. So even if it is bioplastic, even if it is nanomaterials containing bioplastics, they have to be disposed with a proper protocol. Otherwise, it is mere waste of producing any replacement to conventional polymers. So hope you enjoyed the presentation. So be safe and good luck. Thank you.
Thank you, ma'am, for the wonderful lecture and enlightening us on the topic use of natural nanoparticles in food technology. Now we have a few questions in the chat box, ma'am. So
Yeah.
the question is, do certain nanoparticles accumulate in the body and cause biomagnification?
Yeah, of course. See, zinc oxide, we say it is not, it is not pathogenic or it is not harmful. We say it is generally said that zinc oxide has no toxicity, but that is what I said. If when when it is above the optimal limit, when it is above the permissible limit, any nanoparticle can cause, cause disease or hepatomegaly or splenomegaly or any material can cause harmful effects.
Yes, ma'am. And the next question is, can nanoparticles be carcinogenic?
Of course, yes. So in the slide, oh, sorry. So I had shown a slide stating, able to see the slides?
Yes, ma'am. We can see your
Yes, yes,
slide.
ma'am.
Yeah. So here, see. Sahu evaluated the cytotoxic potential of 20 nanometer nanosilver. So everywhere we use nanosilver, but it also has toxic effects. It can also cause cancer. Okay, like Hep2, colon cancer cell lines. It can cause cancerous effects also. So it is one report. There are thousands of reports on cytotoxicity of nanomaterials. It is just for for your information.
Yes, ma'am. Thank you, ma'am. Now I'll call upon Ms. Yurani to give vote of thanks.
Yeah, yeah. Thank you, Alfred. Okay. So on behalf of our organizing team, it's my privilege to propose a vote of thanks on this occasion. And my heart fills with a lot of gratitude and respect for our distinguished guest, Dr. Sirajanusha ma'am, for not only sparing a valuable time, but also for enlightening us with a detailed presentation on nanobiotechnology in food preservation. Especially like ma'am, like when you were talking about right from the beginning, like you were talking about the liposome usage, or you were talking about the quantum dots from there, talking about the detailed composition of the packing material, and one side on the transport phenomena. At the end, you were also giving us detailed information on intelligence packing and toxicity of the nanomaterial. That was extremely good. And I think we here the target audience were like from UG and PG students, and all of them are really benefited. Like you are also talking about the mechanism how it kills, which is there for first year students in microbiology, and this nanomaterial and nanotechnology is there for PG. So I think you had covered for all UG and PG now. So I think the topic was really good, and hope the students enjoy it. And next, I would like to thank our management and our beloved chairman, Dr. Bahu Manoharan sir, managing director, Mrs. Jessie Priya ma'am, and director, Mr. Sasashaya sir, and of course our principal, Vadi Sesha sir, and our dean ma'am, Dr. Parvadavardhani ma'am, for their continuous support in the conduction of such events. I express my sincere thanks to our Professor and Head of the Department, Dr. G. Srikumar sir, for his immense and continuous support in conduction of this event. Thank you, sir. And I also thank Professor Dr. Renuka ma'am for her support in conduction of this event. And next, I would like to thank my staff coordinator, Ms. G. Kalavati ma'am, who is assistant professor in our department, and student coordinator, Alfred Francis, final year, for their tiresome effort in chalking out this event and making this event a great success. Last but not the least, I would like to thank all my dear colleagues and all my students for participation in this event, and thanks for extending your cooperation in the smooth conduction of this event. Thank you. And also students, there are more upcoming BioTalk series in the following weeks, and which this will be communicated shortly. So hope to see you all there. Okay, thank you. Thank you, ma'am. Thank you, Sirajanusha ma'am.